The meeting objectives for this particular meeting is to try to give you an understanding of the current differences in the European regulatory approval and HTA approaches, and discuss the potentials for harmonization and alignment. We want to have forward-facing discussions on decentral cancer clinical trial performances and on the use of digital tools for this purpose. And we'll be addressing the issue of forward-facing discussion on options to accelerate drug development programs in general. And finally, we're trying to stimulate an understanding of the current status of global regulatory collaboration and an open discussion on options for further extension. If we go on to the next slide. The program has been put together by a program committee that I had the honor to chair, but I really need to acknowledge the major contributions of my five co-workers, you might say in this sense. Claudia Hay from Merck Healthcare in Germany, Kim Lyerly from AAADV in the United States, Michael Zayak from Novartis in Switzerland, and last but not least, Roger Wilson, who is representing WeCan, a patient advocacy organization, and Roger is part of the sarcoma advocacy as well. We'll go on to the next slide. Just a little bit of information on the virtual platform that we will be using. There is a question and answer possibility for you and a polling possibility that works under the Slido format. You can touch the button Q&A and then insert a question. And I encourage you to insert questions if you have any while the discussions are ongoing, while the lectures are ongoing. You can already do so. We will obviously try to answer the questions you have in the panel discussion, and the moderators will pick up the questions. If you have any question that you see pop up there that you truly like and forgot to ask yourself, you have the option to put your thumbs up. And then the more thumbs up there will be for a certain question, the more likely it is that the moderators will pick up that particular question to ask it to the panelists. There are other issues here over here and possibilities that are likely well known to most of you. There's a networking possibility under people. You can find the schedule for the meeting on the schedule. If you want to go to the live streaming events, you need to push the button stream. And you can find more information on the speakers and CDDF on the lower two buttons, speakers and CDDF. I think that's enough for this type of information. It speaks for itself. It's an easy to handle tool. If we go to the next slide. The first session is entitled Integration of Regulatory Assessment and Assessment of Reimbursement, and will address the issue of how can we help our regulators that will have two options. One is the regulatory assessment of the dossier that's been submitted, and the other one is a health technology assessment with a completely different aspect, and yet those people have to work with exactly the same dossier. So the question is, how can we improve this? How can we support the regulators in the work that they need to do? It's my pleasure to introduce at least one of the moderators for the session one. Can I have the next slide? Which is Roger Wilson. Roger is a member of the National Cancer Research Institute and the Sarcoma Studies Group. He's a member of the NCRI Consumer Forum, honorary president of the Sarcoma Patients Euronet, chair of the EWARD-C patient panel, and a faculty member of the Methods in Cancer Clinical Trials workshop that is also organized by, among others, EWARD-C. And Roger was an important part of the program committee of this meeting. And Roger will be introducing his co-chair. Roger, the floor is yours now.
Thanks. Good afternoon, everyone, or good morning, wherever you are. As you probably guessed from that, I come from a patient perspective, and as it's a rare cancer patient, I think you'll find that rare cancers becomes something of a focus because some of our speakers have an interest in the rarer community as well. And it's my pleasure to introduce a good friend and long-standing colleague, Professor Mark Lawler from Queen's University Belfast. Mark is a scientist who's got a very strong interest in health data and has been a great innovator in a number of areas, driving innovative precision medicine being a particular interest of his. He's the scientific director of DataCan, the UK's National Health Data Hub, and chair of the International Cancer Benchmarking Partnership, a very significant and very prominent roles in the world of European and UK healthcare. So Mark, over to you to say hello.
A pleasure working with you again, as always. It's always good for us to link up together, and really looking forward to the session today. I think we're going to have a really interesting session, and would encourage you to put questions in Slido, as Jaap said. I'm now going to hand back to Roger, who's going to introduce the first poll.
Right, we've got 3 poll questions for you. So if we could see the first one, please, Michael. Thank you. How likely are you to see significant and publicly prominent change to processes in your organization in the next 2 years? So the choices are, it's already happening. We will see change, significant change, remember, within 12 months. Change is being planned so that hopefully it will be introduced within 2 years, or it's unlikely that we're going to see change in 2 years. So over to you now to click and answer. I'm seeing 100%, 50% shared. I'm sure more answers will come in and change this. 50% shared. We're on thirds now. Good. Yes, the bottom curves are moving only slightly, and the already happening line is holding at about, or between 60 and 70%. So that's very heartening. Thank you. Let's close that one now, Michael, and move on to question 2. The following terms may be used to describe your research drug development approach. Please rank them in order of priority. So the first one we suggest is safety-centric, the second one product-centric, the third one market-centric, and the fourth one patient-centric. So rank in order those 4, sorry. I'm not sure that that's the right slide. Safety-centric and patient-centric are currently equal. Patient-centric is now in the lead. Market-centric is at the bottom. It's very interesting. I suspect that being a patient, as I am, patients might look at that evolving shape and argue with it, and sometimes suggest that perhaps the truth is product-centric rather than patient-centric. But that's a patient viewpoint. Interesting that safety is second. I like that. So thank you. Thank you, Michael. Can we go on to number 3? Right, please consider the following statement. To become patient-centric, our research needs to actively involve patients at every stage in the research process. And the choices are strongly agree, need time to think about it, disagree, or strongly disagree. And a very quick 100%, suggesting that a couple of people are coming in very quickly. It's not moving, it's staying at 100%. Someone on a little more moderate. and no one is disagreeing at the moment, which is nice to see. Involving patients at every stage in the research process is, of course, quite a challenging thing to do. You know, I've been privileged to be able to do it a couple of times with industry partners, and I think it's a really valuable thing for you to achieve and a challenging thing for patients to do, but enjoyable and satisfying. So those numbers are very, very heartening. I'm glad that people strongly agree in general, and overall there is agreement from everyone that's voted. So thank you. I think we can call that a day there. Let's hold that there then. Thank you, Michael. And now back over to Mark.
Thanks very much, Roger, and very interesting results from that poll. And as Roger knows, I'm also very keen on patient centricity and also co-creation and the principle of empowerment of patients. So we hope that everybody, the 84 participants who are currently on the session, also ascribe to those viewpoints. It now gives me great pleasure to start the formal aspects of the session in relation to the talks, and it gives me great pleasure first to introduce Professor Carla Holleck from the Amsterdam University Medical Center. And Carla is going to talk about adaptive pathway development, a clinical approach in Fabry disease. Carla, the floor is yours.
I'd like to thank the organizers for asking me to talk about an adaptive pathway development for one of the lysosomal storage disorders, Fabry disease. First of all, I will show you my disclosures. I'm involved in pre-marketing research with pharmaceutical companies, and I have also been part of the platform Medicine for Society, which is part of the Amsterdam UMC over the last couple of years. So although it is a very specific topic that I'm going to talk about today, I would like to tell you a little bit about the background of lysosomal storage disorders, which makes you understand why we at a certain point developed a pathway like this, or a scheme for a pathway. Now, what you see here on the right-hand side is a cell. And as you probably know, the cell has a garbage disposal system to keep the interior neat and tidy. And for that, we need the lysosomes, which can digest macromolecules. And within the lysosomes are many enzymes, as you can see them here. And they have the ability to catalyze reactions to break down macromolecules. If there is a defect in one of those enzymes, there will be a buildup of storage material, which will then result in a lysosomal storage disorder. Now, there are many enzymes, so there are also many disorders, and you can see here some examples of them very, very differently, not only in between the disorders, but also within the disorder. That means that the phenotypic diversity is enormous. So on the top, you can see, for example, Gaucher disease, a very severely affected child, but also a nearly asymptomatic octogenarian. And the same holds true for almost all of the lysosomal storage disorders. I'm telling you this because it's important if you want to develop a treatment strategy, which I show here as the first one for Gaucher disease. One of the enzymes that is defect there is glucocerebrosidase, and you see here Professor Roscoe Brady, who developed the enzyme, who discovered the enzyme defect in the 60s of last century, and also helped to develop the enzyme replacement therapy. He's injecting a child with the enzyme as we speak here on the figure. And the effect has been very, very successful. You can see on the right-hand side that there is extreme organomegaly in these patients. They have huge spleens and livers, and that is reversible. But also, there were not only dramatic effects, but also dramatic prices. And already in these early days, there was some criticism about this high price of the drug, which could be more than $300,000 per patient per year. Now, after this success in Gaucher disease, that has paved the way for the development of several other enzyme replacement therapies. And if you look in one of the reviews, you can see that people find these enzyme replacement therapies in general to be safe and effective for several of the disorders, including Gaucher, but also Fabry disease, mucopolysaccharidosis, and Pompe disease. But I think we should have an open eye for the real world situation. And that situation is quite different because, in fact, treatment is not a cure, not for any of the disorders, even not for Gaucher disease. And as I said, the phenotypes can be extremely variable. Also, that would probably mean that the natural disease progression is also very variable. And that is a challenge to say when or with what dose a patient should be treated. Because the effect probably would depend on the timing and the disease severity. So open questions are, what is the window of opportunity for treatment for each of these disorders? And also, if it doesn't work, does it mean that early treatment would work? That has not always been proven. And another important issue is that screening initiatives emerged after these treatments became available, revealing many patients with very, very mild phenotypes that maybe do not need any treatment at all. So for these open questions at the time, we already found it necessary that there were more independent collaborative efforts for an appropriate use of these therapies. And we were very fortunate in the 90s of last century to receive some funding, some public funding from the Ziekenfondsraad, which is currently the National Healthcare Institute, to do studies on dose finding, biomarkers, long-term complications for Gaucher disease, where we could establish, for example, that individualization of treatment is quite possible. And it's not just possible, it also is very effective and it saves a lot of money. The disorder that came after Gaucher was Fabry disease, which is quite a different disorder, although the stored lipid is quite similar to what we see in Gaucher disease. The storage is in other cells. It is in vascular wall cells, in the heart, in the kidney, so it has more of a vascular type of expression with renal failure, strokes, cardiac failure, acroparesthesias. You can see here a boy who's comforting his brother who has very painful acroparesthesias in his speech. Here are the typical features of the face of a Fabry male and a thickened heart that you can see in Fabry disease. It is an X-linked disorder, so from that only there you would expect large differences between males and females, and indeed that is the case. So if we look at the disease course in Fabry disease, in this graph, you can see, for example, that in non-classical, that means females with a very high residual enzyme activity, they can be completely asymptomatic. Well, we also have a group of males that have some residual enzyme activity and females that come from a severely affected family. They have a similar disease course, and then we have the classically affected males. And as I already gave away a little bit, that relates very well to the residual enzyme activity and also the amount of storage material. And we could measure storage material in the plasma as a very good biomarker and indication of the severity of the phenotype. Now, enzyme therapy has been developed for Fabry disease already more than 2 decades ago. And interestingly, there were 2 preparations that were developed. Agalsidase alpha, which was then studied in a short clinical trial and received market authorization in 2001. And because it was a conditional approval, there was a registry set up by the company for that. But at the same time, there was Egalcidase beta from another company that had at the same day a market authorization under exceptional circumstances with another registry. Both of these enzymes came with a price tag of 200,000 euros per patient per year. But still, as in the other disorders, we could see that there were many open questions. So what is the dose? What patient do we need to treat? At what time should we treat these patients? These were all questions that were not resolved at that time. Nevertheless, in our country, because of the high price, after some years, people wanted to know whether there was a cost-effective treatment. And we, with support by Zonneweg, performed an HTA study based on our Dutch cohort. And we very carefully selected untreated controls from the historical files that were very well matched with our treated patients. And we could figure out that there was indeed some effect, especially some delay in treatment, and the treatment gave some delay in the development of complications. On the right-hand side, you see a little bit of the scheme of the Markov state transition model that we developed with Marcel Dijkgraaf. And because of the effectiveness being limited with that high price, the cost effectiveness analysis showed a very, very high price per quality, between 5.5 and 7.5 million. So that was at the beginning of that kind of discussion on what a life should be worth. And there was a media uproar because the Healthcare Institute at that time had a preliminary advice to stop the reimbursement of enzyme replacement therapy, not just for Fabry disease, but also for Pompe disease in our country. And as I said, that rose some questions like, what's a year of life worth? And so that sort of set the scene that these kinds of treatment should be monitored better, and that we should also have the ability to sort of more stratify our patients and more selectively give that treatment. Now, as I said, there were some obligations from the EMA to set up registries to also look at the effectiveness on the long term. We had the Fabry Outcome Survey from Shire and also the Fabry Registry from Genzyme. And as I will show you, these registries were very well managed, but they were also lacking a lot of data that were important to answer this kind of question. So again, with the help of Zonneweg, we received some funding to set up an independent registry in our GGG project. And you can see the title here, and we were able, in collaboration with centers from London, Würzburg, Canada, and our center, and support from the patient organization, to set up a database with almost 600 patients. So in our database, we had data on both of the preparations. The phenotypes were uniformly defined. We had full records of clinical symptoms for almost more than 5 years. And importantly, we also had biochemistry outcomes that would help us to look at effects based upon surrogate markers. Now, our PhD student, Maarten Arends, is shown here, who could, for the first time now, very clearly show that the natural course of patients, so untreated patients, was very, very different between classical males and on the one hand side and non-classical females on the other side. So the proportion of patients with major clinical complications you can see on the left-hand side is very, very different. And also, for example, renal dysfunction, which is a major risk factor for development of future complications, and also a major indicator that treatment is not going to be effective anymore, is in fact only seen in classical males and only in a very, very small proportion of other patients. So that of course means that you need to stratify your patients. So what I show here is what has happened between 2001 and all the years after. Initially, we thought it was potentially effective for all patients. Then it's perhaps not effective in advanced disease, maybe not in patients with cardiac fibrosis or renal failure. Then here, the studies were done that it is not cost effective. We also found that antibodies may interfere and that there are certain risk factors for non-responders. In the meantime, there were screening studies that revealed many non-classical patients. Well, we have found out, and others as well, that non-classical females have a very benign course. So it took 20 years, as you can see here, for a good independent consensus on start and stop criteria based upon independent studies. And I think this is an important background where we came from to say that we could do this differently. So maybe with adaptive pathways to patients, you could develop a strategy that you could gather evidence over time and then have also progressive licensing. Well, this has been already launched in, I believe, 2015 as a proposal by EMA. And it requires an active early dialogue with regulators, but also HTA, patients, healthcare professionals. And very importantly, you need to use real-world data to supplement the clinical trial data. And the principle is, of course, to have an iterative development. So initial focus on a narrow patient population and then perhaps expansion to a wider population. But as I said, you need also a very good way to collect real-world data. What we did in our study is just develop a sort of theoretical scheme that could have been followed for Fabry disease, which was published in Drug Discovery Today by Yvonne Schiller. And what you see here is, I think, what all of you know as the traditional scenario. So pre-marketing consultation is especially between industry and regulators. Then a clinical trial is performed in a restricted population, in this case, mainly in classical males. And then there will be a conditional marketing approval at one time point. And after that, the effectiveness is in the hands of industry in different drug registries, where the evidence generation can be very, very difficult. And results are reported to the EMA and not directly to healthcare professionals or to patients. So there is no alignment of these parties, and that will result in continuing doubts on cost effectiveness, which may in the end hamper patients' access to medicine. So the real world experience sort of is not connected to what's happening in this situation here. So we have, based upon the principles that were already set out by the EMA, developed an alternative scheme, an improved scenario that would build on those elements with an iterative development where all stakeholders agree at an early stage on data development plan. Trials will be done in patients that likely benefit most. So these could have an early license, and then further studies are needed to see where other patients with other expression of the disease could also benefit from treatment. So that requires a very, very rigid and well-maintained disease registry, which is not for drugs, but for patients. And for that reason, we feel it's very important that such a registry is led by an independent board and already set up in the pre-marketing phase. So hopefully, if you start that, you can also already at an early stage collect data that are important for HTA, so that HTA assessments can immediately be performed at the same time or quickly thereafter when we have a better view on the real world data. So as I said, the registry, an independent registry, is crucial to our. We are convinced that that is very, very important, and we also reported already on that and on the features of such post or authorization registries in the past in 2015. So you can see what we felt from a healthcare professional point of view, what is important as elements of these authorization, post-authorization registries. Another thing that I think we should mention here is that not just the process of approval, but also the process of access could be improved. So on the EU level, there I think could be improvements, but of course we need to realize that payers will also need an assurance that what they are going to reimburse is indeed effective. So for orphan drugs, we are now in the process of developing a scheme or a project that has elements of the Dutch Drug Access Protocol that allows early but controlled access to treatment. And the idea is that you should signal already months before market approval that a new drug is coming to the market, where you can develop a protocol with a rare disease expert center. And then in a dialogue with insurance companies and also supported by patients and of course the pharmaceutical company, come to a sort of scheme where you already can start early on to treat patients on an individual basis. And if this patient proves to have a benefit based on preset criteria, then the reimbursement can be taken over by insurance companies. And over a preset course of time, you can also collect data, not only from your center, but maybe international in a registry, to eventually say whether this treatment is really effective enough to be completely taken up in the insured package. So coming to an end of my talk, the conclusions of what I've shown you here today is that with the increasing number of orphan drugs, we really need new pathways towards early conditional approval at EU level, but also alternative routes to access at the national level. And no surprise, this requires early dialogues with all the stakeholders and a robust system of evidence generation with independent registries, but also at the same time to not have further delays in development of national systems of access. Thank you for your attention.
Thank you very much, Carla. Really interesting presentation, and we'll be having questions at the end of the 4 talks. So we'll come back to you on that, but I will encourage people from the audience to put your questions into Slido. Now hand back to Roger to introduce the next speaker.
Thank you, Mark. Our next speaker is Dr. Dan O'Connor. Dan is a doctor, a cancer researcher, was a clinical lecturer at University College London and the Ludwig Institute for Cancer Research, before becoming in 2006 a medical assessor for the MHRA. I first met him, he was on secondment to the EMA, working in Amsterdam and in London. He currently works at the licensing division of the MHRA, which is now the UK's drug regulator. He has a special interest in rare diseases, in early access to medicines, and patient-reported outcomes. And he's also very actively involved in patient engagement and involvement. So Dan, over to you.
Good morning and good afternoon. I'd like to thank the organisers for inviting me to talk at the annual CDDF conference. Of course, it's a real shame that we can't be together to be able to share some presentations and some discussion, but it is a great pleasure for me to present on a topic called the Innovative Licensing and Access Pathway. And this is a new streamlined approach to both regulation and patient access. So the first slide really is a very important slide actually, because it highlights one of the real key components to the ILAP, which is partnership working. Much stronger working between the Medicines and Healthcare Product Regulatory Agency, which is the UK Medicines Authority, and with the HTA bodies in Great Britain, the All Wales Therapeutic and Toxicology Centre, the AWTTC, NICE, the National Institute for Health and Care Excellence, and the Scottish Medicines Consortium. So this is a real key component in terms of thinking about how the ILAP might bring medicines to patients more rapidly than is normally appreciated. So what is the ILAP? So the ILAP ambition is to deliver safe, early, and financially sustainable patient access to innovative medicines. And as I mentioned, the real key component is this strengthening partnership between the MHRA, the medicines regulator, and 3 UK HTA bodies, NICE, SMC, and AWTTCC. Importantly is patient input. And hopefully throughout the presentation, I'll highlight areas where we are trying to embed the patient input right from the beginning of the pathway all the way through to patient access, ensuring that those products that are developed for patients really have taken into consideration the views of patients as the end user. What we're trying to achieve also with the ILAP is greater alignment in terms of the evaluation and the access activities throughout the whole pathway, with a much more integrated approach to regulation and HTA. It's not only the partners within the partnership, but also we are working much more strongly with the NHS, National Health Service in England and Scotland, for example, but also other really important players in the life sciences ecosystem, such as our Accelerated Access Collaborative, other UK healthcare system partners. So now I'll move on to describe the key components to the pathway. The first part of the pathway, which I'll come on to describe in more detail, is called the Innovation Passport. This is a new medicines designation. It complements another medicines designation that we have called the Promising Innovative Medicines designation of our Early Access to Medicine scheme. But this Innovation Passport designation links to a portfolio of activities around the creation of a roadmap to patient access. This roadmap is called the TDP, the Target Development Profile. And this creates a unique UK roadmap utilizing tools from a toolkit to provide a platform for sustained multi-stakeholder collaboration. These tools are really important. So these tools are there to drive efficiencies, to build on the collaboration between the partners and the sponsors, and to support evidence generation and evidence requirements that are going to be suitable for both the medicines regulator, but also the HTA body. And the idea behind the ILAP is to try and pull together in a much more integrated approach this pathway, bringing in expertise from the different partners in the wider healthcare system, including the NHS, to really serve patients going forward for those innovative products in a way that they can be accessed as quickly as possible. So a little bit more about the Innovation Passport designation. This is the gateway, if you like, to the ILAP, and it enables access to the pathway and those future important activities in terms of streamlining the development and the access pathway through the TDP. There's built-in flexibility with multiple entry points along the pathway. Importantly, a sponsor can apply with non-clinical data, so they can come very early before their first in human studies, or they can come when they already have some clinical trial evidence. And the ILAP is open to both commercial and non-commercial sponsors. We've created a broad and inclusive definition of innovation in order to capture a wide range of products. And this does include drug repurposing. So this is particularly important for those non-commercial sponsors who are in this space. The non-clinical entry point, so before that clinical data, before those first-in-human studies, provides for the long-term ambition for these much more integrated interactions. And we want to think about the patient right from the start. So as I'll come on to the criteria of the Innovation Passport, you can see that that's really a true focus of the Innovation Passport. But what the designation does right at the very beginning, by having this unique opportunity for the MHRA, HTA bodies to have these discussions around whether a passport should be issued or not, really provides right from the beginning a real platform for that multi-stakeholder dialogue. And it is, I think, a really clear example where there is joint decision-making between the MHRA, the Medicines and Healthcare Product Regulatory Agency, and the HTA bodies, but also with patient input. So the decision around whether a passport should be given or not is not only made by the organizations, but also includes, from the summer of last year, patients from our patient and public reference group. And this is a really important point. There are 3 criteria for the Innovation Passport, and I'll now come on to describe them in more detail. The first criteria is that there needs to be a condition that's life-threatening or seriously debilitating, or there is a significant patient or public health need. The second criteria describes what type of medicine is it. Is it perhaps a truly innovative medicine, such as an advanced therapy medicinal product, an ATMP? We've seen a number of exciting developments in the oncology field in this space. Could it be a clinically significant new indication? And here we're perhaps thinking about repurposed medicines. So we know that repurposed development is a really innovative part of drug development. And we are seeing a number of older products being repurposed in oncology field. Perhaps the medicine has been developed in a rare disease or other special populations, such as in the elderly or in the pediatric population, or perhaps the medicine is being developed in line with objectives for public health priorities. The final criteria, and perhaps the most important criteria, is that the medicine needs to demonstrate potential benefit to patients. And this could be either through improved efficacy or safety, so the activity of the product, or a contribution to patient care or quality of life as compared to alternative therapeutic options. So what has the activity in the ILAP been to date in terms of the, in terms of the Innovation Passport activity? So in the first year of operation, so ILAP was launched in the 2021, January 21, there were 76 applications for the Innovation Passport. As you might expect, and this reflects actually the drug development pipeline, oncology was a major contribution of applications, and you can see that there were 25 applications. The next common area in therapeutic area was neurology with 13 applications, and then respiratory. But actually we did see quite a really nice spread amongst different therapeutic areas. And to date, we have granted 44 Innovation Passports, 23 were pending, 8 were refused, and 1 was withdrawn. Importantly, we're seeing interest from a variety of size companies, so the larger pharma, but also the small companies. And in fact, we've seen an application from a small UK university. And more recently, we have actually given an Innovation Passport designation to a not-for-profit research organization. And that's a really pleasing development, which shows that the ILAP is attractive to all of those that are in the innovative medicines development space. The ILAP also does include products that are going to go through the FDA Orbis pathway. This is a pathway run through the FDA, but also includes work sharing with other countries such as Brazil, Australia, Health Canada, Swissmedic, Singapore Authority. And here we've seen 11 applications express a specific interest in this oncology pathway. And there is a good mix of both rare and common diseases. And in fact, our first Innovation Passport, and you can see in the right side of the panel, the press release for this passport was actually approved in a rare condition von Lippel-Lindau disease back in February of 2021. Oops, sorry. So just a little bit more detail around what type of applications we've seen and what stage of development we've seen. And this is clearly of interest because what we want to do is to create really long-term interactions around the trajectory of a product and its development. So as you can see here in the graph, a good proportion of products were coming in the early or mid-stage. So the early stage is defined before the first in human studies. So that means the innovation passport was based on data from a non-clinical model before clinical data were available. The mid-stage, where you can see there were 23 applications, this is before confirmatory studies. And then you can see in the final two bars where the applications have come in more mature setting. But this does mean that there's less input that the partners can perhaps achieve in terms of using the tools of the toolkit. But I do still think that it is an important interaction around discussing perhaps the route to marketing authorizations, thinking about some of the innovative ways that you might consider managed access pathways. But anyway, what we're hoping to see in the future as the ILAP beds in, that we see a shift of the products that we're seeing, the products move much more earlier. So actually we see the bulk of the products coming in the early to mid stage so that we can have these long-term discussions. Now I'll move on to the TDP. So the Innovation Passport designation is the link to this portfolio of interactions around the creation of the TDP roadmap. As you can see on the bubbles on the left and right-hand side of the panel, that there are a number of specific topics or areas that we're really interested to discuss, such as special populations. So this may be women's health, rare diseases, comorbidities. What's the development to date? So what's the approach to the evidence generation? Are there novel approaches that we really need to consider? Perhaps there's a surrogate biomarker that needs to be investigated. What are the challenges in that development program? How can we really streamline and think about really driving those efficiencies in the development program? We know that scientific advice is really important for drug development, so really highlighting the importance of that scientific advice, and particularly that joint scientific advice between the medicines regulator and the HTA body. A dedicated section on patient engagement, so ensuring that the patient voice is embedded, trying to understand what patient engagement the company has already conducted, what patient engagement the company is planning to conduct, but also what patient-reported outcome measures have been included within the development program. What's the strategy for pro measures to support that understanding of how the medicine affects the patient? Product development to date, so really understanding better where the product is in the life cycle, and then understanding whether that product has got multiple indications. So it's not just about a single indication, but a discussion really about that whole approach to that medicine development, whether we can extrapolate data from a previous application to a future application. So really trying to have a much more holistic approach to the discussions that we have, as opposed to the targeted scientific advice that often can be quite narrow. So the TDP, when it's complete, will define the key regulatory and development features for that product, and really identify or try to identify what might be those pitfalls and challenges, and create a roadmap to deliver early patient access, really with the ambition to try to create a product that is as regulatory and access ready as possible to ensure that streamlined approach to patient access. I've mentioned the tools of the toolkit, and this is some of the tools that we have either in development or are readily available. And as you can see, they cross a number of different areas of a clinical development program. So for example, we have novel clinical trial methodology, really important for those companies or sponsors that are considering perhaps basket studies or platform studies or other different types of ways to collect data, particularly surrogate markers or using biomarkers. We have mechanisms to support clinical trial recruitment and a rapid clinical trial dossier pre-assessment service. We also consider center accreditation and adaptive inspections from our inspections colleagues, such as for GMP and GCP. We have flexible licensing routes, so you can see in the middle of the panel with rolling review, accelerated timetables, day 150, using the international options such as FDA Orbis and the Access Consortium. And we also have in the bottom left of the panel, you can see we're called the UK HTA Access Forum tool. And this is a new tool being developed by our HTA colleagues, NICE, the SMC and the AWTTC. And this is a specific access tool really for the HTA bodies to co-discuss with the sponsor about the route to access. And this tool will be launched later in February. So as you can see, this toolkit is currently quite diverse. But over time, we're hoping to add to this toolkit and create more tools as we gain more experience in the ILAP, understanding better what parts of the development program really need assistance and where we can add value into the system to support those innovators. So now my final few concluding slides, just to sum up in terms of what this might mean for individual stakeholders. So most importantly for the ILAP is really the focus on the patients, their families and carers. What does the ILAP mean for them? So earlier last year, we set up a dedicated ILAP patient and public reference group, and this is really to start to drive a much clearer end-to-end approach in terms of embedding the patient voice in the drug development pathway. What we want to achieve to ILAP, which is obviously, of course, very relevant to patients, is safe and timely access to innovative medicines addressing those patients' needs. We want to create a system where patients are at the front and the center, and we've started to do this by ensuring that the innovation passport designation discussions include the patient voice, so whether we grant a designation or not, we'll have the patient there. But we have a specific tool on patient engagement through the toolkit, and we want to develop more approaches to ensuring that the patient is fully embedded in the ILAP process. And importantly, the opportunity for patients to perhaps discuss the design of the clinical trials and what might be the benefit to patients through that product once it's on the market. What does ILAP mean for the life sciences industry? So there's obviously this opportunity, we hope, for these platform discussions. So earlier engagement in the design of innovative clinical trials, driving those efficiencies and really focusing on the patient needs and potentially reducing clinical trial timelines. We want this early platform dialogue to really help us understand what evidence is required from both a regulatory and HTA perspective, and then this obviously helps the sponsor in terms of their planning. Does that data set mean that the medicines regulator can make a decision at a particular time point? And is that data set, that evidence that's been generated, is that also going to be suitable, importantly, for access? As I mentioned in the toolkit, there's the flexible and expedited regulatory routes, which can lead to earlier market access. And this earlier planning really is going to hopefully help to ensure that the evidence developed is going to potentially support managed access and the commercial decisions, with an opportunity to plan at a cross UK national level, with opportunities to also include NHS payers. And what does the ILAP mean for the MHRA, UK HTA agencies and the UK payers? I think what we found has been a fantastic opportunity during the first year of operation has been this opportunity to collaborate much more. We always worked with colleagues from our HTA bodies, but never to such a degree that we've worked during the ILAP. So we can collaborate, we can do our best to align. It helps us do our jobs better by facilitating safe and early and financially sustainable patient access to important medicines, so really clear goals from all of our organizations. It also helps us in terms of thinking about alignment of evidence generation. So when we have all of these discussions together, we can really understand better what each of us needs to be able to make the decisions that we have to do for the future. And also there's a key interest in real-world evidence. So we have one of the tools of the toolkit, which is the continuous benefit risk tool integrating real-world evidence. So really understanding, which parts of the development program have to be conducted with clinical trials, but perhaps which elements could be collected in the future post-authorization through real-world evidence collection. And a key component, and I think this is one of the first, is that we have this joint decision-making around the innovation passport. So we sit in the same room through what we call our innovation passport steering group, and we make a co-decision around whether that innovation passport designation should be given. This common and earlier understanding of the regulatory and access pathways increases the opportunity for alignment And the timely engagement does really allow opportunities to create a much better commercial and managed access approaches where relevant and where needed. So my final slide in terms of just where we're going in the coming year. We're currently working on a new digital offer, which will hopefully have marked improvements in terms of those submitting the applications for the Innovation Passport and the target development profile. In relation to this, but also in our first year of operation, we've learned a huge amount and gained from experience of the applications and modified some of our processes and procedures. So there'll be additional guidance and further information updated on our webpage. We're going to develop some case studies with sponsors to really highlight the benefits of engaging with the ILAP, what we've learned, what have been the important approaches to delivering some of the benefits that I've highlighted in the previous slides. The new HTA Access tool will be released in February, and there's the continuous developments and process improvements of the other tools. And there'll be further collaboration with other stakeholders in the UK life sciences ecosystem to derive this end-to-end approach with hopefully the possibility to help drive efficiencies also in clinical trial delivery. We also want to encourage developers of genomic medicines to enter the pathway, and this is clearly important for those many of those oncology products. And this is because many of these products also rely on in vitro diagnostic capabilities, so companion diagnostics are really important to ensure that we have co-development and co-discussions around the medicines development, but also the diagnostic component. How are we going to find these patients? How are we consistently going to consider the diagnostic requirements for understanding whether a patient should be given a particular medicine or not? And finally, we'll be building on the lessons learned for consideration of a medical devices pathway. Medical devices also offer huge benefits to patients. And so a similar pathway linked to some of the concepts of the ILAP is currently under active consideration. So I'd like to thank the organizers and yourselves for your listening, and I'll be happy to take any questions at the panel session. Thank you very much.
Thank you, Dan. I have to declare an interest. I have been a member of that patient and public reference group that Dan mentioned for the last 6 months. So I've seen the ILAP work from the inside, as well as hearing about it in the way that Dan's explained it. And I must say I'm very excited by all ideas that start to move safe and managed access to patients earlier in the drug pathway. It's really exciting to see some of these changes evolving and now coming through in regulation. So now over to you again, Mark.
Thanks, Roger, and absolutely agree with you in relation to that early interaction and early intervention is really important for patients. To continue the ILAP theme, and I think it's a theme, if you like, of this overall session, Daniel Martin from Merck Healthcare Germany is now going to look at the industry experience with ILAP. Daniel, over to you.
Good afternoon. My name is Daniel Martin. I am a global regulatory leader for the oncology therapeutic area at Merck Healthcare KGaA, and I will share my experience with the process in the registration of an anti-cancer medicine during last year, 2021. I will go first through high-level details of the innovative licensing and access pathway, and then the actual experience with ILAP. Next, please. The innovative licensing and access pathway is a new UK MHRA scheme implemented in January 2021, post-Brexit, which aims to accelerate time to market, facilitating patient access of innovative medicines and or medicines treating areas of unmet medical need. The ILAP enables multiple entry points depending on the stage of development of the product, the data available, the ambition of the applicant to engage with the stakeholders, and appetite for new innovative ways of working. It provides opportunities for enhanced regulatory and other stakeholders' input, such as the National Institute for Health and Care Excellence, NICE, the Scottish Medicines Consortium, the National Health Service England, providing companies with advice on clinical trial design to ensure optimal data generation for both regulatory approval and health technology appraisal. Next, please. Inclusion in the innovative licensing and access pathway enables setting up a target development profile. This is a roadmap defining key regulatory and development features. The inclusion in the ILAP also enables MHRA participation in Project Orbis. That's the collaborative marketing authorization review, which is led by the FDA, and on which relevant agencies that are participating are the Australian TGA, Health Canada, the UK MHRA, of course, the Singapore, Swissmedic, and the Brazilian ANVISA. The Innovation Passport designation is a prior requirement and is acting as the gateway entry into the ILAP pathway. Next, please. The Innovation Passport is the gateway, as I mentioned, to enter into the ILAP pathway. The eligibility requires meeting 3 criteria, and it is critical meeting all 3 of them. Criterion 1 is that the medicine is treating a life-threatening condition or a condition with significant unmet medical need. Criterion 2 has several different points that can be met. Just one is required to meet criterion 2. For instance, that the innovative medicines, that it is an innovative medicine such as an advanced therapy or a new or chemical or biological entity, or a new drug device combination, or that it is a clinically relevant new indication, or that the medicine is treating a rare disease or a special population, or that development is aligned to the objectives for public health priorities of the Chief Medical Officer. So only just one of these needs to be met at least to meet the criterion number 2. And then for criterion 3, the MHRA is considering that the medicinal product has the potential to offer benefits to patients. Next, please. In order to progress the Innovation Passport, then a submission of an application for MHRA is required. The company is then invited to meet with MHRA to discuss how their product fulfills the 3 criteria. Following the meeting, the partners, MHRA, NICE, Scottish Medicines Consortium, jointly consider if the criteria have been met. The company is informed of the outcome within 4 weeks. And after Innovation Passport designation, there is the option to progress the target development profile that defines key regulatory and development features and identify potential pitfalls, creating a roadmap for delivering early patient access. Next, please. With regard to the practical aspects of the ILAP pathway, I will cover 4 aspects. A brief introduction, the MHRA interaction, the aspects related to the marketing authorization application, and also those of the health technology appraisal, and finally a high-level summary of the applications submitted to MHRA to date. Next, please. So the ILAP enables multiple entry points into the scheme, depending on the stage of development of the product, the data available, etc. In our case, we were in an advanced development stage, the marketing authorization application almost ready for filing in December 2020. So that was just about the scheme going into effect. Brexit timelines ruled the scheme initiation being 1st of January 2021, and obviously due to the legal reasons of UK being a member of the European Union before, the scheme could not come into effect earlier. The ELAP guidance was made available in December 2020. That was very shortly before the scheme initiation. However, the MHRA was flexible and available to guide applicants within the extent of the information available. The ELAP allowed us for additional interactions with MHRA assessors and also gave us positive expectations for an improved health technology appraisal process. Next, please. In the context of the MHRA interaction, the ELAP pathway enabled additional interaction with the MHRA assessors, providing the opportunity for additional discussion, the identification of pitfalls and weak spots in the planned marketing authorization application. Also, it allows us a better understanding of the assessor's expectations on defined topics, and also provided us with the opportunity to give additional clarification on quality, non-clinical, and clinical aspects of our innovative medicinal product, as well as an opportunity for the assessors to get preliminary familiarity with this new product ahead of the filing. Next one, please. With regard to the practical aspects of the marketing authorization application and the evaluation, the earlier interactions with the agency enabled us building a more robust application. The scheme opened the possibility of a rolling review, which in our case we did not use as our dossier was almost ready for filing. However, it's a positive feature for the pharmaceutical industry. It opened as well the possibility to participate in international collaborative assessments, such as the project Orbis, led by the FDA, as I mentioned before, as well as the accelerated assessment of the application with a 150-day procedure, which we used, and the time frame actually was met. Overall, we consider that the features of introduced by ELAP for the marketing authorization application were very positive, and they offer us very positive benefits, actually. Next one, please. With regard to our experience with health technology appraisal, the Scottish Medicines Consortium has introduced additional process for ELAP designated medicines, which provides an interim reimbursement. However, in England, the NICE process seems unchanged, and in our case, with no apparent benefits from being ELAP designated, especially taking into account the main objective of ELAP of facilitating patient access of innovative medicines and or medicines treating areas of unmet medical need. So overall, the benefits from health technology process have been less significant than those on the marketing authorization of our evaluation in our experience. Next, please. And finally, to conclude my presentation, I would like to mention that during the first year in place, from January to end of December 2021, there were 71 ELAP applications received by the MHRA, with 41 innovation passports awarded. That is 85% over the applications assessed. 7 innovation passports not awarded, which account for 15% over applications assessed, and 22 applications in process. This shows a great success of the scheme, actually. Thank you.
Thanks very much, Daniel. Really interesting and really very complimenting Dan's presentation earlier, but interesting to see the industry involvement. And yes, would agree, definitely the take-up is exceptionally good. Now pass on to Roger, who's going to introduce the last speaker in the session.
Thank you, Mark. Statistics has never been a strong point of mine, but it's always interesting discovering a statistician and a leading thinker in statistics who can explain things nice and clearly. And our next speaker is certainly that. Mark Beiser is, he holds degrees from Brussels University, from Cranfield, and his doctorate from Harvard School of Public Health. He founded the International Drug Development Institute in 1991. And he's worked with EORTC and the Dana-Farber Cancer Center in Boston. He's founder of Clue Points, which is dedicated to statistical monitoring of clinical trials, an intriguing subject area. And he's an associate professor of biostatistics at Limburg's University in Belgium, working on statistical methods for personalized medicine. So moving the statistical analysis of randomized trials from a single endpoint to a patient-centric benefit-risk assessment is, for me, an intriguing topic. So Mark, over to you.
Hello, I'm going to talk about the statistical analysis of randomized trials, and I'm going to discuss a specific issue about this analysis, which is moving this analysis from a single endpoint to a patient-centric benefit-risk assessment, so a much more global assessment of the benefits and risks of a treatment. So when we do the statistical analysis of randomized clinical trials, as statisticians, what do? We actually use so-called conventional statistics that you've all seen. We calculate survival curves, we compare survival curves, we calculate forest plots for subset analyses, we compute hazard ratios, odds ratios, and all kinds of statistics. And if you think of it, these analyses have two problems. The first problem is that we focus on typically a primary outcome measure or a primary endpoint, for example, overall survival and the hazard ratio of overall survival for the comparison of the two treatments. And in fact, the second problem is that not only do we focus on a single endpoint, but we produce statistics that are very hard to understand. A patient typically does not really understand a hazard ratio unless that patient is a statistician. But we prefer to talk about single, simple statistics of the primary endpoint of the trial because that makes our life easy. Now, if we go to the patient view of a clinical trial, what matters to them? What matters to the patients? Well, patients have several concrete questions, such as how much longer can I live? Or will treatment be painful? Or what will be my symptoms? What is the chance that I will do better on treatment than on control? I mean, the last question here is probably more a question, again, that a statistical patient would ask themselves. But so these questions relate to the benefits of the treatment, the harms of the treatment, or the risks of the treatment, and then some kind of an overall assessment of the benefit risk. And typically, we just don't do this as statisticians in clinical trials, working in clinical trials. And that is probably something we should be doing more and more. So I'm going to show you a short video. It's a 4-minute video that will show you how we can actually position a statistical approach of the patient-centric view of randomized clinical trials. And this video is pretty simple. It's not statistical at all. It's aimed at patients or clinicians or non-statisticians. And as you will see, it is fairly easy to understand the approach that this video is advocating. And I will come back after the video to show you some details of the statistical analysis we propose.
Advances in research and new technologies mean that the number of new treatments approved by the authorities has increased tremendously over the past couple of decades. Having a larger choice is nice, but how do we make sure that the best possible treatment is given to a certain patient? When new drugs or treatment methods are proposed, we study them across several phases of clinical trials that help identify the most promising approaches among those considered. Finally, The best of them is compared with a currently accepted standard treatment by administering them in 2 groups of patients. These trials are designed in such a way that this comparison is based on the treatment only, while minimizing the influence of other factors, such as age, gender, or underlying diseases. Typically, the trials focus on a single outcome of the treatment considered most important by the researchers. In practice, however, a patient might be concerned by a different or even several outcomes of the treatment simultaneously. A treatment that gets rid of an infection but is very toxic is not the same as a treatment that fights the infection and is less toxic, right? The method of generalized pairwise comparisons, or GPC, allows to do just that, to evaluate all outcomes of interest at the same time and in a desired order of preference. Consider a trial that was performed to compare appendicitis treatment by antibiotics rather than the usual surgery. We begin by asking ourselves a question. Among the possible outcomes affected by this treatment, what are the most important for us personally as a patient? Then write them down on a list starting from the most important. We then take 1 patient from the antibiotics group and 1 patient from the surgery group and compare their outcome of the highest importance on the list. In our case, the number of days it took our patient to recover. Then we decide if the patient on antibiotics from this pair has recovered faster, or maybe the patient after surgery had faster recovery. Sometimes the decision is not easy to make. Maybe both treatments have taken similar time to work. Then we would look at the next outcome on our priority list and compare its value. Can we decide if antibiotics were doing better or worse than the surgery here? If we can, we leave it at that. If not, we would go and look at which of the 2 patients had better quality of life 1 month after treatment. All of the patients on antibiotics are compared in this way with every patient from the group undergoing surgery. We can now measure the effect of the antibiotic treatment compared with surgery. This measure is called net treatment benefit. It reflects the difference in the number of times that the treatment with antibiotics was better and the number of times that it was worse than the surgery. A value larger than 0 means that the antibiotic treatment is better. If the net treatment benefit is less than 0, we would choose the surgery. This conclusion is based on the particular preferences that we have chosen. What if someone else had a different order of personal preferences? The net treatment benefit value will then change. It will inform which treatment is preferable based on this new list of personal choices. Without GPC, your treatment decisions are based on general expert consensus on what constitutes a single most important outcome, one which may not necessarily reflect your preference as a patient. GPC is a method that empowers you by allowing to participate in making treatment choices. Now the decisions are truly tailored to your own wishes and needs.
All right, so this video showed you the essence of the methodology that I would like to discuss today. And I'm going to go into some details of what was shown very clearly, much more clearly than I could present it in the video. So what the method consists of is to define thresholds, but before we do this, define priorities. As the video said, each patient has their own set of priorities. They might, for example, on this slide, choose time to death or survival time as their first priority outcome. And then they might say, well, avoidance of pain may be the second outcome. And then having or not having certain symptoms might be the third outcome, or the severity of the outcomes might also be important. And so we can decide on the list of priorities depending on the patient preferences. Now, an analysis would be done using that particular order of preferences. Secondly, the patients, in fact, don't just think as to the order of the outcomes or the priorities of the outcome, but also what kind of difference would be important for them on each of these outcomes. For example, in terms of time to death or survival time, what kind of difference in survival time would be relevant? It's obvious that if the survival of a patient was improved by 2 days, that would be trivial. That wouldn't be worth taking a treatment. But if it was 2 months, or even better, 2 years, then it would become really interesting to take the treatment. Likewise, for pain, the intensity of the pain is as important as having it or not having it, and the same for symptoms and so on. So not only do we list the outcomes in a pre-specified order, but we also define for each outcome a threshold of clinical relevance. And having done that, then we can use a statistical method. You might think, well, this is all becoming very complicated, but we can use this method of generalized pairwise comparisons. And that, in fact, as I will show, makes the analysis so simple that it's just unbelievable. You can do so much with so little, in fact, mathematics. What you do is as it's shown in the video, is to form pairwise comparisons. So you take one patient in the treatment group, one patient in the control group, and you, for example, here denote by Xi the outcome of the patient in the treatment group, and Yj the treatment of the patient in the control group. And in fact, then you decide if that particular pair of patients is a win, if it favors treatment, or a loss, if it favors control. If you look at survival times, for example, if Xi is greater than Yj, in other words, if the patient treated has a longer survival than the patient in the control arm, then that's a win. The patient treated is a win. Otherwise, it's a loss. If the treatment in the control group does better than the patient in the treatment group, that's a loss. And if the two survival times are equal, then obviously you can decide, and that pair is called a tie or an uninformative pair. So that's the pairwise comparison. Now what we do is to do this process on all possible pairwise comparisons. Here I've shown an example of two groups of patients, two groups of 6 patients. So 6 patients treated in blue, 6 patients in the control group in red, and you form all the possible pairwise comparisons. 6 by 6, it's a 36 pairwise comparison problem. So you have 36 pairwise comparisons. For each of the pair, you decide if that pair is a win or a loss or a tie. And then the calculation of the net treatment benefit is fairly easy. You just do the calculation of the number of wins. You do the calculation of the number of losses, and you know how many pairs you've got. You just do the subtraction of number of wins minus number of losses, divide by the number of pairs, and that's your net benefit. So that's, in a sense, the expression of the overall benefit for all outcomes considered, if you have several outcomes. And it shows how many patients are actually doing better on treatment than control. It's the probability that a patient taken at random would do better on treatment than control. If the net benefit is 0, that means there is no benefit. There is no difference between treatment and control. If the net benefit is 100%, that means all patients do better on treatment than control. And if the net benefit is minus 100%, obviously that's an undesirable situation. That means the control group would uniformly do worse than the treatment group. Of course, that would not be a situation that we would want to be in. Now, in practice, of course, the net benefit is never plus or minus 100%. It's, something around 0, but sometimes it diverges from 0 if the treatment has an effect in a way that is interesting, both statistically and clinically. Now, what makes this method particularly attractive is this business of the threshold of clinical relevance. When you compare the outcome of the first patient with the outcome of the second patient, you can decide that the pair is a tie for if the difference in that pair is less than the threshold of clinical relevance. So, for example, for survival analysis, you could say, well, if the difference between the two survival times is less than 3 months, let's say, then I would consider that difference is really not clinically important. If the difference is more than 3 months, then of course it becomes important. And I can do the analysis with different thresholds just to see what kind of threshold would make the treatment significantly better than the control. And the other extension that's very important is that we can do this for multiple outcomes. If we have multiple outcomes, as I said, we can order them by descending order of priority. And then we look at the first outcome. If that first outcome allows us to classify the pair as a win or a loss, we're done. If not, so in other words, if we don't have information in the first pair, the first outcome makes this an uninformative pair or a tie, then we have to go to the second outcome. And the second outcome might then help us to classify the pair as win or a loss. And then if we have more than 2 outcomes, we continue this process until we've exhausted all the outcomes. And at the end of the day, we may remain with a few uninformative pairs, obviously, but all the other pairs will help us to decide which of these 2 treatment arms, the control or the treatment, is in fact better. So the method is conceptually extremely simple. Mathematically, it is also extremely simple. The characteristics and properties of this method have been studied in great detail, and the advantages of this method are, in fact, many. The first advantage is that this is a completely non-parametric approach. It doesn't require any assumptions. You often hear about assumptions we have to make statistically, such as the survival distribution has to be exponential, the hazard rates have to be proportional, or the distribution of the outcome must be normal or something like that. We don't need any of this here. It's completely non-parametric. We can use this method for multiple outcomes, providing we can order the outcomes by descending priority. And the outcomes, of course, can be efficacy outcomes or safety outcomes or any other outcomes. Could be quality of life, could be even biomarkers if we wanted. So we can really throw in this methodology any outcome that we can measure in a patient. The GPC also permits a mathematically rigorous benefit-risk analysis that takes into account the correlation between the outcomes. The benefit-risk analyses that you see performed do not, in general, take the correlation between the outcomes into account. And that's a huge problem, because think of a treatment that's very toxic but helps a few patients. That's a very different situation than a treatment that is very toxic in the patients who benefit from it, rather than in the patients who do not benefit from the treatment. You would want to use a treatment that's toxic, providing it would help the subset of patients who also have toxicity. But you wouldn't want to use this treatment if it induced toxicity without helping the patients who have the toxicity. So that correlation between the outcomes is extremely important to do a correct benefit-risk analysis. The net benefit is a universal measure of the treatment effect that has a straightforward probabilistic interpretation. It's the probability that a patient will do better if treatment than if on control. So it's a very simple probabilistic interpretation of the net benefit, regardless of the outcome or number of outcomes you look at. It's always the same interpretation. And last but not least, coming back to the title of my presentation, this is intrinsically patient-centric because the order of priorities can be adapted to each patient, and the thresholds of clinical relevance can be adapted to each patient as well. So let me mention a few applications of GPC that we've been working on recently. Of course, we've used this feature of thresholds of clinical relevance, for example, to say, well, is the survival on treatment better by at least 6 months compared with a survival on control, or at least 12 months, or at least any number of months? So we can vary the number of months and look at the net treatment benefit as a function of the threshold that we choose. We can also look with GPC at non-standard situations. One issue that has bothered clinicians recently is the fact that immunotherapy in oncology produces a treatment benefit, but which is delayed. So you have to begin treating the patients and expect to see a treatment effect later, sometimes much later, sometimes several months later. And this is because of the mode of action of immunotherapy. It takes time to be activated, in a sense. So the standard methods of statistical analysis, in particular the log rank test, the Cox model, and all these methods that we use in these situations are just not appropriate because they don't account for the fact that the treatment effect is delayed. So in that case, GPC can be a much better option. We can say, well, I want to have a survival time of at least so many months, and that will in fact compensate for the fact that the treatment benefit gets delayed in time. Another situation that I think is quite important is when you have two events that are time related, for example, progression of disease and death. Typically what we do is the time to first event, so time to progressive disease or death, whichever comes first. The problem with this analysis is that if the patients progress, we ignore the time after progression until death. And of course, that's really putting things upside down, because what we would really like to do is to look at time to death. And for patients who haven't died yet, for those patients, we look at time to progression. That would make much more sense clinically and also for the patient. So that's another cool application of GPC. Mixed outcomes, like I said, sometimes we can analyze outcomes that are not the typical traditional times to event or binary outcomes such as proportions of events, but we look at things like circulating tumor DNA. You can easily add circulating tumor DNA levels in a GPC analysis. The type of event that you look at can be any, and that's really very powerful. Another application is multidimensional scales for quality of life, for example, where you have multiple dimensions that are measured. GPC can be used, again, to get an overall assessment of the quality of life on multiple scales, which is not easily done with any other method. And this can be pushed to the limit, and there are a few researchers who work in that direction, using very highly multidimensional data, such as gene expression profiles, which you can also analyze with GPC. So it's a very kind of promising method, and I'd like to finish with looking at who might be interested by GPC. And, you know, again, if I come back to the title of my talk, I believe that it's the patients who might benefit most from a GPC type of analysis. And I've indicated here by a circle plus something that's really important. A threshold of clinical relevance is really important for the patient. And mixed outcomes, benefit and risk, is very important to the patients. And I've listed a few other users of GPC, potential users: clinicians, regulatory agencies, health technology agencies, or health technology assessment groups, the pharma market access group in the big pharma industry, and the pharma R&D departments for the regulatory approval. So if we kind of group these different users together, we have the consumers, in other words, the people whom we want to target GPC at. We have the regulators, of course, who have to review a new drug application and approve a new drug or not. And then we have the payers or the producers, typically the pharma industry that produces new drugs and that want these new drugs to be used and approved. So I think GPC actually can be useful for all of these groups for different reasons and for different types of applications. But again, if you look at the patient column here, it's obvious that the GPC approach provides patients with a much better and much more exhaustive view of the benefit risk of a treatment, rather than having to rely on separate analyses of each of the endpoints using statistical notions that are typically quite complicated to explain, and certainly for patients who are not mathematically trained, very difficult to understand and to interpret. Thank you very much for your attention.
Thank you very much indeed, Mark. I found that presentation really fascinating, and I really admire the way you're approaching a problem which patients themselves have been articulating for many years, and challenging the way we're moving forward with science. Let me hand back to Mark, who's going to now introduce the way we're going to address the panel discussion.
Thank you very much, Roger. So just invite all the speakers back into the session, so we can now go into the Q&A and the panel discussions. So we're going to take a mixture of questions that we have ourselves prepared, but also questions that are coming in from Slido, and keep those questions coming in. But we're just going to start off with some questions that both Roger and myself will ask. So first of all, Carla, a great talk, really enjoyed it. Obtaining high quality long-term follow-up information on participants can be difficult. How did you achieve that within your academic registry?
Yeah, thank you, Mark. That's a real great question. And I think it's one of the most important obstacles that we now have, that you need to set up a kind of database or registry to capture those data. Well, for our studies, we have been able to work with 4 dedicated centers, and we had a PhD student who really visited as a kind of monitor the centers and set up that database. And actually, we are now doing a similar thing for metachromatic leukodystrophy, which is a physician and patient-driven registry that ideally in the end should be aligned, of course, with regulators and pharmaceutical companies and payers. And then you can collect data on different therapies for one disease. So this is something that is for rare diseases should be set up at a European level. But you need structure and financing to really set that up and make it sustainable for the future. But I think it's doable. We are working with payers as well to get that kind of structure and finance from the ground.
Really like that idea, and also the fact of looking at different registries and bringing them together, particularly in rare diseases. And I'm sure that will resonate with Roger as well. One more question for you, and then I'm going to switch over to Roger. One of the things you emphasized in your talk was really the great importance that you place on real-world data, particularly in the context of, as you described it, iterative drug development. And also the role of registries, as you've already highlighted there, for describing and articulating different patient pathways. Question I'm going to ask, and I might also come to Anne in relation to this, do you feel the regulators pay enough attention to real-world data?
Well, I think they are paying attention to it, but it is still on a very sort of academic level. And with our idea is that you should make it concrete. Just start with a registry, show that it's going to work, see where the obstacles are, try to solve those. Because there are several pathways that already have been developed or that are being talked about that use real world evidence, but I haven't seen lots of those being really effective. So I think it could be done, but you need a very robust, as I said, structure, but also financing and monitoring, because you want high quality data. I think that's very important. So several people say, well, those registries and real world data are not sufficiently powerful because they're not robust and it's not adequately monitored, and there's differences between centers, etc., etc. You can resolve that. I'm absolutely positive that we can do such a thing.
Thanks, Carla. Anne Williamson, I'm going to bring in. Anne Williamson is from EUNet HTA from the Netherlands. Anne, from your perspective in relation to real-world data, what are your thoughts?
Yeah, I think it's a very critical perspective, and I think it really begins also with having a good and early on scientific advice between the developer, the regulators, and also the HTA side, and because you need to get those questions on straight very early on, and because otherwise you also will not know what you're following up on in those registries. And I also think that really the European collaboration is key, although probably you might have different needs from a registry on a national level, but you also need to have a broad set of patients there. So I think we need to collaborate. Might not be easy, but we really need to go there.
Thanks very much, Anne. Now I'm going to pass over to Roger, my co-chair.
Thank you, Mark. I'm going to pick out three of the questions in the Slido for Dan O'Connor. Because they relate to one another, and it's about the global issues and the global relationship that MHRA will have and probably needs to have with ILAP. The early access to medicines scheme is resource intensive. One of our delegates points out, when you're applying for global market authorization. So how will ILAP align, Dan, in terms of submission requirements in the global sphere? And is there experience, someone else asks, of how an early ILAP involvement can be incorporated into a global development program? And our good friend John Smith asked the question, will linking with Orbis slow you down? So three views on the global issue. How do you respond?
Thanks, Roger, and thanks very much for the questions. So I'll start with EAMS, that maybe not everyone is familiar with EAMS. So EAMS stands for the Early Access to Medicine Scheme that we launched in 2014. And a bit like one of the programs that you heard earlier from Carla, it aims to fill a specific gap for patient access when maybe clinical trials are fully recruited, and there's no opportunity for patients to access important medicines, and patients maybe cannot wait 6 to 9 months because their disease would have progressed sufficiently. So we've understood and we've heard from feedback from companies that EAMS can be resource intensive, and we ran a public consultation back in the summer of last year with the intention to create a legal basis for EAMS. And part of that legal basis will be to reduce regulatory burden, which will reduce regulatory burden from a supply perspective, but also for collecting real-world data. So we're creating a new framework to collect real-world data with hopefully much more of an end-to-end approach with managed access. So hopefully EAMS will become less resource intensive, but we do understand, particularly at the time of marketing authorization, that there's a lot going on, and applying for EAMS can be an additional challenge, but hopefully in the coming months we'll solve some of those issues. So how will ILAP be different? So ILAP is actually positioned through the entire development program. So you've got a lot less condensed, if you like, interaction, but it's over a longer period of time. And so the goal of ILAP is to have that sustained early discussions with both the regulator and the MHRA. So in terms of submissions, I mean, that's probably a target development profile, perhaps on an annual basis or every couple of years for those early stage products, which would be around scientific advice using the tools of the toolkit. So difficult to compare, but I would think that probably ILAP, because it's spread out over a much more longer period, that it's going to be less resource intensive. In terms of the second question around, are there any early experiences in terms of ILAP influencing global development programs? We've heard from some sponsors who've come for the roadmap meetings or the target development profile meeting that's really been an important discussion. We fully recognize that virtually for all conditions, that there is a global development plan, there is a global approach to commercialization of the product. And ILAP, I think, can provide a useful safe harbor discussion point for discussions around difficult, challenging areas of opportunities to drive efficiencies for clinical trials and other surrogate markers. And these will be applicable across the globe, obviously with discussions with colleagues in other regulatory authorities. But as I mentioned in my presentation, we hope to develop some case studies with sponsors to really demonstrate some of the added values. And we take this point on board about globalization. So it's a good hook for us to kind of consider when we're looking at some of these case studies. And the final question, I think, was from John, which I've momentarily forgotten. And Roger will have to remind me. Sorry, Roger.
Yes, it's about linking up with Orbis. Is that going to slow you down?
With Orbis, yeah, great. That's right. Thanks. So thanks, John, for the question. So yes, so as part of the ILAP, it's mandatory for companies to engage with ILAP for Project Orbis. So I don't consider that would slow us down. I think we've seen great collaboration through that platform with colleagues from the FDA and the other countries. What it does do, though, because it's linked to the innovation passport and hopefully that discussion through the target development profile, it does really allow for that early horizon scanning between the medicines regulator, but also the HTA bodies in terms of what products are coming through, and particularly what are those potentially routes for markets. So if it's going to be an international opportunity, then obviously we know that, but there were clearly other flexibilities in our innovative and flexible licensing tool of the toolkit around the day 150 and the rolling review approach. So obviously the company will weigh up what are the particular opportunities and benefits of the different routes that we offer for marketing authorization. Thank you.
Thank you, Dan. I'd like to actually extend that question to Daniel Martin. Daniel, do you feel that ILAP and the requirements you have for getting authorizations more globally, they do align and work together effectively?
I think so, actually. When you look at the requirements that the MHRA have, they actually overlap with the requirements that first-tier agencies have. So FDA, Japan, basically would have similar set of requirements for the similar schemes. So in Japan, you would have Sakigake, or in the US, you would have some of the schemes as well for products that have an unmet medical need or that provide a significant advance in science. And I think in our experience, the criterion or the criteria followed overlaps very much.
That's good. That leads to the sort of fairly obvious question for you, Daniel. Do you think that ILAP does make things easier because it coordinates so well, or because it's deliberately accelerated? How do you feel it is better? Is it better?
For us, actually, in the setup of Brexit, where there was a new requirement to get products registered in the UK, unlike in previous months, the iLab actually provided a scheme that helped us to bring an innovative medicinal product to the patients faster. We had our product already in a very advanced stage of development. Nevertheless, with the iLab scheme, that enabled us to have additional interaction with the health authorities, go into accelerated assessment, which actually was under our radar, and also potentially access other schemes, like it's been mentioned before, Project Orbis. So it's up to the applicant whether it wants to go or not. But it adds additional flexibility in my experience, it added additional flexibility for us in our path, and it brought speed in order to bring the product to the patients.
Good, thank you. Back to you, Mark.
Thanks, and I want to bring it to the voices of the patients. Dan, you mentioned in your presentation about including the voices of the patient, but I'd like both yourself and Daniel, and probably Anne as well, just to tease that out a little bit more and say how were they facilitated, and what were the sort of opportunities, but also the challenges. So it's sort of a multifaceted question. Maybe start with Dan first.
Yeah, thanks, Mark. And it's great that people have picked up on the patient aspect. I mean, it seems to be a real theme from this session, so it's really good to hear. So when we launched the iLab, I mean, when we looked at the criteria, and as Daniel's mentioned, I mean, the criteria are similar to maybe other jurisdictions in terms of what we might want to see in terms of prioritizing particular products or indications. But one of the things that we were really keen to do from an agency perspective, but also from the iLab partners, to have a complete sea change in the way that we involve patients with the way in our decisions, but also in some of the other activities we do, such as scientific advice. I think we had, as an agency, we had some really good examples of patient engagement involvement, but we didn't really offer it in a way that was particularly systematic, let's just put it this way. So it was more ad hoc. And what the iLab does is I think it provides a really visible framework to hang off a lot of those really important engagement activities. So the first, as I've mentioned, is that we set up, and as Roger's mentioned, he's part of this group, we have a dedicated patient group called the iLab Patient and Public Reference Group. It includes a number of different patients and patient representatives from a variety of different conditions and areas. And that group is really helping us in terms of evolve the iLab in a way that we want to see this end-to-end engagement with patient engagement. So we've started with having patients in the room for the discussions around the innovation passport. In our guidance to sponsors, we're encouraging very strongly for sponsors to include information from their patient activities in the application for the innovation passport. So a kind of a pull and a push, if you like, in terms of that patient involvement. And actually we have seen that. We have seen patients coming to some of the innovation passport meetings with the sponsors, and they've provided some really excellent interventions around, for example, a new rating scale. I mean, how a change in the rating scale, how valuable is that change to patients? So these are incredibly helpful for us to hear as regulators. We have in the TDP a dedicated patient engagement and involvement section, which, as I mentioned, covers off really, all of the aspects around that we want to know is how much engagement has the company already conducted with patients? And this is something that's actually often missing from the regulatory knowledge, because we know that sponsors do engage a lot with patients, but there's just nowhere for companies to provide us with that information. So, for example, you really have to proactively ask for that to find it. And so that's why the TDP really tries to call out those activities. But again, through the TDP and the enhanced patient engagement tool, we will have a much stronger strategy for involving the patients. We've not got it right completely yet. I think there's still a long way to go, but I think we've really earmarked some key elements where we want to improve our patient involvement. And I think the iLab offers us an unparalleled opportunity and a very visible opportunity for us to do that.
Thanks very much, Dan. I'm a big fan of the TGP, so no, absolutely agree there. And Daniel, from your perspective, just from the industry perspective, what are your thoughts in relation to that facilitating and empowering of the patient viewpoint?
So in that case, in my case, from a regulatory perspective, we don't see that as much as possibly health technology appraisal or during the registration process is not so obvious to us. But what was very clear is that the iLab process, it added, as I mentioned, the flexibility. And depending on your stage of development, you can enter the scheme earlier. So those companies, ideally, that have an early candidate already that can look into the scheme. In our case, we were close to filing, and also the scheme allowed us to apply and go through. And I think that was that flexibility actually was beneficial for us.
Thanks very much. And Anne, from the HTA perspective?
Yeah, I think if we look from the experiences of UNEDA, so the European Network for Health Technology Assessment, there are definitely a few areas we can tackle with patient input and what is relevant and what we learned. So we tried to come up with methods that we could use for both the joint scientific consultations or the early dialogues. We heard a couple of names for this, but also for then the actual assessments to have patient input at different stages and see where it is most relevant. So if I focus a bit on the HTA aspect, because I think we heard a lot already on the early dialogue side, we do need to remember that within UNEDA, we separate a clinical assessment that we do within UNEDA versus then the actual appraisal that is done on the national level. So your actual decision and the conclusions that you make regarding added value and reimbursement, that's done nationally. And we at UNEDA then provide the summary report. So if we keep that in mind, I think it also brings a different need and perspective of why we want to bring in the patient input and how it is helpful. Because for us, then it was much more relevant to have it at time of the scoping. So what really is your research question and which outcomes do we need to really analyze it with? And that is where we found patient input to be most relevant. Whereas if you do it more on the appraisal level, I think the patient input also really needs to be included when you make the decision for reimbursement to really capture the impact that it could have or will have if you not reimburse it. However, we really did learn that not every HTA agency is as far advanced when it comes to patient input, and organizations really needed to learn what does it mean to bring in patients, how do you then do that, how do you identify those, how do you make sure it's understandable what we ask, and also how then do you translate the input within an assessment report. And that I think is still one of the biggest, well, maybe not a challenge, but that is something we really need to still work on to also, after we tick the box, we did gather the input, what do we do with it then and how do we present it? And to me, that also means that in that earlier stage, through the early dialogues or the scientific advices, you really very carefully document what the patient input was and also what then the most patient relevant outcomes are so that we can tackle those in the actual assessments.
Thanks, Anne. And that brings me nicely to my next question, and to bring Mark in. One of the things, not only patient engagement and involvement, but obviously also the potential for patients presenting data, and I'm particularly thinking of patient-reported outcomes. And if I remember from your talk, Mark, one of the things you highlighted is there seems to be a methodology that would actually encourage such a move. Would you maybe like to tease that out a little bit more?
Sure. Yes. The question specifically was about, doing sort of GPC analysis, using the technology that I explained to compare not just two treatments, but more than two, because obviously for a patient, oftentimes there is a choice between alternative therapies. And it's a really good question. One thing I did not explicitly say during my talk is that you need to have comparable groups. I mean, you can't, compare real world evidence from some patients to the data from randomized clinical trials, that wouldn't be a valid comparison, obviously. Whether you use GPC or any other method, that comparison would be intrinsically biased. So I sort of placed myself, without saying it, but I placed myself in the context of a randomized clinical trial where you have two comparable groups of patients. And the problem, I think, that I see, at least with traditional drug development, is that typically drug companies test their own drug with a control, with a standard of care. And then another company will test their drug with the same standard of care, hopefully the same, sometimes a different standard of care. So you end up with all these pairwise comparisons, but that doesn't give you a comparison of the new drug from one company versus the new drug of another company. And of course, that's a kind of a missing direct comparison. I think there should be more trials comparing competing drugs because they may have different safety profiles, they may have different efficacy profiles, they may differ in a number of ways, and that comparison is almost never done. So what I would plea for is to have either more post-registration trials comparing drugs, head to head, or even better, having these platform trials that we now see, upcoming platform trials where in fact you have multiple drugs being tested versus one or multiple controls. And the really nice feature about platform trials, in my view, is that not only will you have the comparison of each drug versus control, but you may even have comparisons between competing drugs, and that would be enormously precious information that we never had in the past and may have going forward if platform trials are more and more used.
Thanks, Mark. And given that ILAP has been one of the themes of the session, I'm now going to try and link it into orphan drugs and bring Carla in. So, just in relation to ILAP, do you think matching orphan drugs to that type of approach would actually be possible?
Is that a question? Sorry, was that a question for Carla? Okay, good.
For Carla, yeah.
Yeah, thanks for that question, Mark. Of course, I think it's very useful for orphan drugs. Still, I'm struggling a little bit, but how it would work in practice. So I also put a question in the chat that what I am sometimes missing is the dialogue, the early dialogue between the healthcare professionals, so the experts on a certain disorder, and also the pharmaceutical industry and regulators. And I think some of the issues that can also be resolved could be with early engagement of healthcare professionals. So for you, it might seem obvious that healthcare professionals are involved. But as far as I'm concerned, I've seen several developments passing with endpoints that really, for example, did not make a lot of sense. And sometimes these trials have failed simply because of the endpoints not being adequately chosen. And then also the entire post-marketing issues. What we have seen is that there could be quite different roads to, for example, for exceptionally, for products that have been authorized under exceptional circumstances, that at some point are fully authorized, but in the real world setting, we are still struggling with lots of questions. So I think that alignment of stakeholders should also involve the healthcare professionals at a very, very early stage. But that also poses a question about conflicts of interest, because some of the healthcare professionals are very tightly attached to pharmaceutical industries. I'm not against that. Of course, I think we should collaborate, but we should be very, very careful in how, for example, people have been supported with their research by the pharmaceutical company to be absolutely sure that what they are going to give and advise is scientifically valid. I'm very sorry that I'm a little bit perhaps cynical about this, but I've seen that in my own surroundings that these things might happen. And I think this is something that is not as yet frequently emphasized as something that we should bear in mind when we are trying to collaborate at an early stage. So I'm not sure whether that this answers your question, Mark, but I also wanted to bring that point forward.
No, absolutely, and really important to bring that point forward. And I think we all agree that that early engagement is critical. I think it's come up in pretty much all of your presentations, and also in some of the comments that both Roger and myself have made. And also, I think very important in relation to, that we need to look at fair value for everybody, for the patient, for the healthcare professionals, for the researcher, and for industry, but they need to be that fair value principles employed. I'm going to hand back to Roger now to continue the Q&A.
Thank you, Mark. I can only echo those words, and I'm a great believer in early engagement with patients. Apart from anything else, it's ethical. Let's be honest. Can I return to Mark Beiser? Mark, you first presented this concept, the GPC concept, more than 10 years ago. So I'd like to know how you're getting it received by the cancer clinical trials community. And, I'm asking this reflecting in my mind that the probability theories of the Reverend Thomas Bayes were developed in the 18th century, and it's only now that they're beginning to be more widely used. And one of our questions touches on a similar subject, asking, does the GPC have the potential to completely replace current statistical ways of establishing the benefit-risk balance of new drugs and indications? Reflect, please.
Thank you for the question. It's of course a very good one, and I think the answer is no. I don't think that this method is aimed at replacing all the good analyses we did in the past. And in some cases, it's obvious that what we did in the past was perfectly okay. I mean, take a very serious cancer, for example, pancreatic cancer, to take one example, where survival is so abysmal. It's obvious that a new drug that has any chance of succeeding in pancreatic cancer really must have an effect on survival, or else some other amazing effect on quality of life, perhaps. But it's a fairly clear case where a single outcome might be appropriate. I'm not saying that it should be always overall survival, but a single outcome might do the trick in that disease. But in many, many other diseases, the assessment of the disease is multifactorial. And if you look at one outcome and ignore the others, you risk, in fact, rejecting good drugs simply because the primary outcome has been missed in the registration trials. Or vice versa, you might actually have a drug that modifies the primary outcome, but has so many other problems, toxicity, side effects, adverse events of all kinds, that in fact, it's not going to be used in practice, or perhaps it will be used against the patient's will. So I think sometimes you need this particular approach, sometimes you don't. And so I don't think that, this approach is always going to be the primary analysis of a trial. But it's very helpful, even as a secondary analysis, to after you've shown that the drug works on the primary outcome, then you can have a more overall assessment of the drug effects, both good and bad, using a GPC approach. So I do think that it has potential to shed light on the benefit risk of probably all new treatments.
How are you getting on, though, with getting this message across?
Well, actually, to be honest, I published this paper that I showed 10 years ago, you're right. And I feel really sorry that I didn't work on this topic more actively than I did. I was kind of looking at other types of things. But since we've come back to this, to be honest, we've received funding here in Belgium to develop this methodology. And I'm really grateful to the public health authorities here in Belgium for funding this project, because we would not have been able to do it without their support. So now we have a team of people working on this methodology. We've done a lot of publications on the properties of this methodology. So I think it's going to pick up now that we've established it has the sort of right mathematical properties. You don't want to establish a method that's not completely validated, in a sense. And now we are at the point that it's been validated, and we try to find applications for it. And it's not very difficult to find applications for it. But yes, it's taken time indeed for this method to become more, let's say, more centrally used than it was before. And in fact, it's been used in cardiology. In the US, you may have heard of a statistic called the win ratio that was proposed by Stuart Pocock also about 10 years ago in 2012. And that statistic is actually used in some cardiology trials where, again, the idea is to look at the time to most important event in patients who have a cardiovascular problem rather than time to first event. So exactly the sort of idea that I developed here. And so that's another application of the methodology. If you have multiple outcomes, look at the time to the most important outcome first, and then the time to the other outcomes second, third, etc. And that makes a lot of intuitive sense. I mean, in retrospect, we almost wonder, why didn't we do this before? And simply because we didn't have the methodology.
Yeah, thank you. I really like that. I think that answer also applies to, I think, some of Thomas Bayes' ideas. Why didn't we do that before? And we didn't really understand the methodology he was putting forward. And I'm not sure that we still do quite, but we're getting there.
Well, this one is simpler than Thomas Bayes.
I'd agree.
Posterior probability. That's kind of hard to understand. I think this one isn't.
I fully agree with you, which is why I'm so attracted to what you said. I'd like to sort of extend what you've said and to Dan O'Connor. And Dan, have you yet seen anyone put forward a GPC approach in terms of looking towards a marketing authorization study? And how would you respond, do you think, if you did see it?
Thanks, Roger, and great talk, Mark. Just quickly, I'd like to just cover off some of the comments from Carla, if I may, because I think they were important ones. So, I mean, absolutely, we want to involve healthcare professionals, and I think regulatory authorities often mix with healthcare professionals because the companies and the sponsors bring key opinion leaders when they have scientific advice meetings. And I think I would say that probably at least 50% of the scientific advice meetings I do, that there are experts from hospitals or academic centres that are brought in a collaborative way by the sponsor to the scientific advice meeting. So it's really important. Through the TDP, we've recognised this, and we actually have a section within the TDP of the ILAP that asks the company to detail what key opinion leaders the company has engaged in their development program so far, so we can get a handle of what's the landscape in terms of the involvement the company has had with different colleagues. And I think the real-world data aspect is absolutely crucial. I mean, if you are going to collect that real-world data, it can't be an afterthought. And so I think that's part of the reason why we see some of the criticism of the regulatory authorities perhaps not engaging with real-world data as perhaps they might do. But it's a little bit like patient-reported outcomes. If they're added in as a sort of afterthought or not really thought really clearly through, then the regulators' hands are actually tied, because it's very difficult for us to actually do anything, because it's either too late for us to make anything meaningful, or we've really got to try to catch up in terms of being able to deliver that. So I just wanted to cover off some of those points from Carla. So no, I think Mark is presenting some really excellent work. I'm not a statistician or methodologist, and we have a great team at the agency who support that. And I haven't actually seen the GPC myself yet in scientific advice, but I think it offers great opportunity. And I think, through the TDP or other interactions, other healthcare authorities, I think having some of these discussions about where this particular methodology may add value, I think is really important. And I think your table, Mark, was fantastic in terms of really identifying, which kind of stakeholders are really going to be the ones that have got most interest. But as regulators, we are, and I'm not just talking about the UK, I'm talking across the globe, that regulators are really interested in new methodology. And often we get a phrase that I always bring out when I can is that, the regulators wouldn't like it. And then you say, but have you asked the regulators? And then you get the question back, well, no, actually we didn't. So just a plea that if you are thinking of new methodology, if you are thinking of using some of the ideas and approaches that Mark has highlighted, then come and talk to your friendly regulators, because we would really like to hear what you want to propose. Thank you.
The same question is obviously now has to be targeted at Anne, because you're also wearing the regulatory hat in this. How do you respond to the GPC ideas?
So I wouldn't classify ourselves as regulators, because we really do the HTA side. But also personally myself, I haven't seen it. I'm also not aware it has been used in any of the UNEDA assessments so far during Joint Action 3. I think we would be in a bit for a surprise if we would see the method coming in. So I'm not quite sure how we would deal with it, because I think we also need to fully understand what consequences might be if the method was not done properly, or whether it was not appropriate to the situation we're in. So we might really need to then have those questions also with a clinical expert to see if it makes sense, yes or no. But I think if this method really is coming and it sounds promising, it sounds also really for the patient choice. So once it already is on the market, really beneficial, then I suppose we really need to have a proper method for the HTA side, or a guidance for how to do the method and how to assess it. So I think that's a bit of work before we are confident enough to really give it a strong placement, because you really base a decision on it. So I think we need to have a bit more confidence and understanding in it first.
Yes, I can understand that. Can I just, it's quite logical now to sort of bounce over to Daniel Martin and ask him the similar question, because obviously he's coming at it from now a different direction. He might be the person we're asking to consider using GPC, and from a patient viewpoint, it's certainly something that I'm going to be taking into mind when I talk to my research colleagues. So how would you respond to the idea of this new methodology? You're on mute.
We're open in the industry to new approaches. I personally have no experience on it, but definitely we would welcome and evaluate new approaches in the drug development field.
Good, thank you. Do you want to comment on that as well, Carla, or you want to bypass on that?
Yeah, well, I have a question about it, because
please.
I was wondering that if you want to make out whether there is a win or a loss, or whether it is a tie, you need to be quite sure about the outcome, right? So for many of the new drugs that come to the market, there's still lots of uncertainty. So how could you, with your model, deal with that, with uncertain effects? Is that possible at all, or is that, in that case, your model wouldn't work?
No, I think, again, the model only uses outcomes that have been pre-specified and then everybody agrees on. So outcomes that are, appropriate for this particular disease that you are interested in. So you would define a list of outcomes, and these outcomes would have to be measured, for this methodology, just as for any other methodology. Are you referring to situations where the outcomes themselves have been questioned because they are unreliably measured, or situations like that?
Yeah, well, when I combine my experience that I just talked about, when you have a heterogeneous group and you don't know whether a patient will have a benefit, and for example, if it is a very chronic disorder, where you cannot look at hard endpoints like death or life and things like that.
Yeah.
It might be very, very challenging, I think, to use that kind of model.
I think you raised two questions, really. One is the uncertainty you have about the outcomes. I mean, in some diseases, some outcomes are very difficult to measure, and they come with, reproducibility problems and stuff like that. That, of course, would affect the analysis, just as it would affect any other analysis, actually, right? But then the other question you raise is, I think, an important one as well that I didn't mention, is if you have a stratified population, in other words, a heterogeneous population where you have very different patients, and the course of the disease differs substantially between these patients, then you would have, just like you explained, in your talk, you would have to really stratify your population between different types of patients. And in your pairwise comparisons, you would want to stratify the analysis as well, such that you match patients. When you do the pairwise comparison, you only look at similar patients. So you would not want to match, I don't remember exactly how you call these patients, but a typical male to a female, or, I don't remember the exact terminology, but you see what I mean. You wouldn't want to do the comparisons across these strata.
The interesting thing is, if I may just add to that, some of that knowledge came after we had this medicine on the market, and that is not an exception when we are talking about rare diseases. So there's a lot of interest, and all of a sudden, in that rare disease, and you will find that the spectrum of phenotypes is much wider than you have seen before.
But that's true of common diseases too. Huh? I mean, I'm interested in breast cancer. It's amazing that breast cancer doesn't exist as an entity anymore. It's, every subtype of breast cancer has to be looked at on its own. So again, this is, in my view, precision medicine. The fact that you look at a specific subset of patients and what do they need. This again is the same problem in GPC as it would be for any other analysis.
Thank you, and thank you, Carla, for that. Mark, back to you.
Thanks very much. Okay, now what I'm going to do is I'm going to take you forward to February 2024. There's been 99 attendees at this session, so really popular session. Jaap is so happy with it that he says, let's do this again in 2024. And he says, those two guys named Mark and Roger, they didn't do such a bad job. Let's have them to chair the session again in 2024. And what I'm going to ask you is, how do you show me that you've actually progressed this area in the last 2 years? So tell me, each of you, in 2 minutes, what you've achieved that actually has really moved this forward for the benefit of patients. And I'm going to start with Anne.
That's always a very lovely question. So within UNEDA, we are now in the phase where the new HTA regulation is being implemented. So I hope by then we're really into a position that we can start working on that one and that we have a fully functioning and well thought of process from the beginning to the end. So really from your scientific advice through your assessment to also registries wherever possible into a reassessment, because I think that's important then as well, that we really do something with the output there. So I hope that is what we have achieved with a world of collaboration between all the stakeholders, but also being still critical in all the methodologies that we're using, not just pushing everything on real world data, because I think also with a proper RCT and with having the proper questions in mind and the outcomes, I think we also can come a long way. So that really would be my hope and a goal for the future. So collaboration, life cycle approach, and also being critical about the evidence.
Super. Daniel?
From my side, working in regulatory affairs basically is what I would highlight is the collaboration with the agencies from earlier stage at a global level, actually. That enables or facilitates the process to bring in medicines to the patients.
Thank you very much. Mark?
Yeah, I mean, I think, to me, what I would hope in the next 2 years is that the agencies, the regulatory agencies in the HTA, work that comes afterwards becomes more aligned, because I think at the moment a drug is approved on very, very little information, on its primary efficacy endpoint, and then it goes to HTA assessment, and sometimes that differs substantially. That's why some drugs are approved and never reimbursed, right? In Europe, it's a real issue. So, I mean, why are these drugs approved if they never get reimbursed afterwards? That means the benefit risk of these drugs is insufficient. So what I would hope is that in 2 years, the methodology for looking at the data from randomized clinical trials, improves, and that there is some alignment from the beginning. And of course, that implies patient involvement from the beginning, such that the endpoints we look at make sense and are the ones that will make a difference in real life for the patients who have the condition.
Thank you, Mark. And really important point there in relation to reimbursement, which is something we touched upon earlier, but I'm really glad you brought that up. And that's because, as you say, it's not much use if the drug is approved, but not reimbursed. That doesn't help the patient at all. Carla?
Yes, well, I think one of the most important things from the viewpoint of the healthcare professionals is that we need to start ourselves to be more proactive in setting up databases, registries, to look at what is going to evolve in the field, what new treatments are going to be developed, and anticipate on those and already start making that kind of registry. So as I said before, that we could also in the future be able to have a registry that also fulfills HTA requirements, for example, and then we can collaborate with different companies and also do some of the comparative trials that we've just learned, maybe not a traditional trial, but also to compare outcomes in the future. So I think we as healthcare professionals need to be absolutely more aware of this. That needs also education, because lots of my colleagues do not know a lot about the regulatory side of medicine development. And another thing is that when I ended my talk, I said we are working on a model with payers to develop a pathway for early access. So if you have a marketing, that doesn't mean access. So I think there is a lot to gain for both patients and healthcare professionals, but also for pharmaceutical companies, because that is really a trouble, as we heard.
Okay, thanks, Carla. Dan.
Yeah, thanks. I mean, 2024 seems a long time away, doesn't it? Thankfully, after the last 2 years. I think within 2 years, 2 or 3 years, we're going to see some of the early stage products that we've already given innovation passports come through to patient access, hopefully. So, what I'd hopefully be able to be presenting is what did we do differently? So was there greater alignment between the medicines approval from a regulatory perspective and the HTA body? And I'm talking about both from a time perspective, because we know that sometimes there's a gap between when we give a drug license and the HTA approval. So we need to try to align those much better from a temporal perspective, but also in terms of evidence generation. Are we sharing, the same types of data? Are we singing from the same song sheet? And how can we integrate some post-approval commitments that we often want to see, particularly in the areas of rare diseases? I want to be able to say that we really have truly matched our ambition for patient engagement and involvement. We started to do it through the Innovation Passport designation, which is a co-shared decision with the HTA bodies, the medicines regulator, and the discussions with the patients. But I hope we'll be able to demonstrate much further integration of patients towards that. And I also hope that we'll be able to demonstrate that we've been really pivotal or supportive of some novel methodology. Maybe it's the GPC, I don't know. Maybe it's some other biomarker or surrogate marker that we've really had those discussions through the novel methodology toolkit and really tried to bring, advancements in terms of regulatory science to patients. So I think there's a lot that hopefully I'll see in 2 years, but maybe we'll have to wait and see. Thank you.
Thanks very much, Dan. Thanks to the 5 of you for showing us your vision of what's going to happen in the next 2 years. If all of that happens, I think we'll all be very happy and we'll all have smiling faces. We started this session with the words of a patient. It's only appropriate that we end this session with the words of a patient. So I'll hand over to my co-chair and colleague, Roger, to sum up.
Thank you, Mark. And if I was going to express my wish for what would be there in 2 years' time, it would be patient involvement. And don't forget that when you involve patients, you have to bring them on, you have to train them, you have to create a fellowship with them in order to get the best from them. I would like now to close the session. I would pay particular tribute to my co-chair, Professor Mark Lawler, a long-standing friend and a delight to work with you again, Mark, so thank you. And a very special thank you to our 4 speakers and to Anne Williamson too for joining us for the debate. We return at 15:45 CET, so I leave you to do your own time zone adjustments. Before you go, can I ask you to remember that the BRELA tool that we're using for this event allows for individual networking. Go into the networking panels on BRELA and you will be able to make one-to-one contact or one small group contact with people whom you might never manage to meet otherwise. So please use the tools that we've got and see you at 3:45. Thank you.